Good morning to all. Computational biology is an interdisciplinary field which combines biology with computer science to analyze and interpret the biological data. It all started with the discovery of DNA, the molecule of inheritance, in 1953 by James D. Watson and Francis Crick. It revolutionized the world of science and technology. Later, the Human Genome Project, an international collaborative mega research program which completed in 2003, this 13-year project of NIH revealed haploid human genome is about 3 billion base pairs long and contains about 30,000 genes. It produced 750 megabytes of data. Genomic, the study of genes and their functions, from this beginning of determining the genomic sequence led to the study of function of numerous genes, functional genomics, to compare the genes in one organism with others, comparative genomics, structural genomics like one. Now, the omics revolution started to characterize, making the researchers to collaborate between different fields. Omics technologies provide the tools needed to look at the differences in DNA, RNA, protein, and other cellular molecules between species and among the individuals of species. It established an open approach to data sharing and open source of software, thereby making the data resulting from the research accessible to all. Genomics, proteomics, and metabolomics of microbes, plants, and animals have revolutionized many fields of science, including microbiology, virology, infectious disease, agriculture, animal husbandry, plant biology. Moreover, deeper knowledge of human sequence variation has begun to alter practice of medicine. The Genome Project has inspired subsequently large scale of data acquisition initiatives such as International HapMap Project, the Cancer Genome Atlas, the recent Human Brain Project, Emerging Human Proteome Project, etc. It opened many avenues in biology, medicine, technology, and computation. Now, biological data are exploding both in size and complexity. This extremely large data can be analyzed computationally to reveal patterns, trends, and associations, especially relating the medicine and agricultural science. Bioinformatics and computational biology possess great potential to identify diseases, determine treatments, and help to make human life better. With this inspiration and knowledge of computer science, fields such as gene technology, medicine, and healthcare can evolve from curing individual patients to healing entire populations across the globe. It took decades to develop vaccines for smallpox and polio and other deadly infectious diseases. Translational computational biology helps to tackle this pandemic COVID in few months. A new era in the science of vaccine developed. Today, we are very proud to introduce our alumni distinguished scientists from the field of computational biology. Dr. Shivamogga Darshan, web application developer from University Alumni, Birmingham, USA, going to speak on understanding cancer through the eyes of big data. Another distinguished scientist, Dr. Nagesh K. from Syngene, Bengaluru, going to speak about computation for genomic data analysis. Dear participants, enjoy this feast of computational biology. Thank you.
Thank you so much, ma'am, for briefing about today's webinar. Now, I request our college principal, Dr. H.T. Krishnamurthy, sir, to speak a few words about the webinar.
Good morning to everybody. Chief guests, participants, and my dear colleagues. DVS College of Arts, Science and Commerce as proud to say that most prominent college in our Malnad region has always tried to its best to get updated in possible ways. This institution is for enrichment of the students and faculty. As a part of this, today I am delighted to notice the Department of Zoology with the collaboration of Computer Science and Alumni Association of our college, under the aegis of IQAC, has organized one-day international webinar on one of the relevant issues, prospects in computational biology. Thus, I here take this opportunity to congratulate Dr. Geeta and Professor Sudhakar, Netravati, and all the organizers to making this webinar grand success. The main thing is that our today's webinar speakers, Dr. Darshan and Dr. Nagesh, are the distinguished alumni of our college. ದರ್ಶನ್ ಮತ್ತೆ ನಾಗೇಶ್ ಮುಖ ನೋಡಿದಾಗ ನನಗೆ ಗೊತ್ತಾಯ್ತು ಇಸ್ ಎ ಫೆಮಿಲಿಯರ್ ಫೇಸ್. ಕೇಳಿಸ್ಕೊತಿದ್ದೀರಿ ಮತ್ತು ನೋಡ್ತಾ ಇದ್ದೀನಿ ಅನ್ಸುತ್ತೆ. ನಾನು ನಿಮ್ಮ ಕ್ಲಾಸ್ ತಗೊಂಡಿದ್ದೀನಿ ಎಲ್ಲ ಗೊತ್ತಿಲ್ಲ, ಬಟ್ ನಿಮ್ಮ ಮುಖ ಪರಿಚಯ ನನಗಿದೆ. ನಮ್ಮ ಕಾಲೇಜಿನಿಂದ ನೀವು ಹೋಗಿ ಬಹಳ ದೊಡ್ಡ ಸಾಧನೆಯನ್ನ ಮಾಡಿ ಇವತ್ತು ಮತ್ತೆ ಈ ವೆಬಿನಾರ್ ಮೂಲಕ ನಮ್ಮ ಜೊತೆಗೆ ಸಂಪರ್ಕದಲ್ಲಿರೋದು ಬಹಳ ಸಂತೋಷವಾಗಿದೆ. ಮಿಸ್ಟರ್ ದರ್ಶನ್ ಅಂಡ್ ನಾಗೇಶ್, ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್. ಗುಡ್ ಲಕ್. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ.
Thank you so much for your best wishes, sir. I invite Prithvi ma'am, Assistant Professor of Zoology, to introduce the speaker of session 1, Dr. Darshan S.C.
Good morning all. It's my privilege to introduce our alumni and pride of DVS College, Dr. Darshan S. Chandrasekhara, as a resource person of today's webinar. He pursued his degree from DVS College with BZCS combination in the year 2002 to 2005. He has completed his master's degree in bioinformatics from Kwampu University and received his PhD from Manipal University. Presently, Dr. Darshan S. is working as a postdoctoral fellow in the Department of Pathology of University of Alabama at Birmingham, USA. Dr. Darshan has co-authored 33 publications featuring in peer-reviewed journals such as Neoplasia, Oncology, Nature Communication, and he has received Best Poster Presentation Awards in many national and international conferences. Currently, his research involves the extensive analysis of cancer omics data to unravel molecular mechanisms evolved in tumor formation and invasion and to identify novel targets for cancer therapy. His main achievement is towards the development of analysis platform, and he has designed web portal that is Alcan or University of Alabama Cancer Database, which is very popular among cancer researchers to study gene expression, methylation and survival patterns in tumor subtypes of 33 human cancers. Developing user-friendly web-based tools with interactive and advanced graphics to facilitate easy retrieval and analysis of public cancer genomics data for medical and non-medical researchers. Alcan has been visited over 4 lakh times by cancer researchers from over 100 countries and cited over 800 times. With this, I welcome back Dr. Darshan S. Chandrasekhara to his college. Thank you.
Thank you, Prithvi ma'am, for inviting our guest. Now, over to you, sir.
Thank you. Thank you for the very kind introduction. Good morning to all. Before I begin my presentation, I would like to first thank the organizers for providing me this wonderful opportunity to present my research to the students and faculty of my own college and from my own hometown. So I was actually a 2005 student, so yeah, it's been a long time since I actually interacted with the faculty and the students of DVS College. So today, my talk is basically about stressing or trying to tell you what are the important roles of computational biology in cancer research. My talk is titled as Understanding Cancer Through the Eyes of Big Data. And I was told that this webinar is attended by the faculty and the students from diverse backgrounds. So accordingly, I have actually organized my talk into 3 major sections. So I'll begin by briefly explaining about the cancer, its basics and biology, and then I'll try to introduce the concept of big data and try to give you brief information about the computational approaches that are available and that we use to actually handle the big data. And finally, I will briefly describe a online cancer data analysis tool that we developed in our lab at University of Alabama. So let's begin with the cancer. So while I was preparing for this particular talk, the first thing that came to my mind was to look into the statistics, the cancer statistics in India and in US. So the latest number that I could get basically tells that in 2018, India had witnessed about 11,50,000 cancer cases, which has led to around 7,80,000 deaths in 2018 alone. And the latest cancer statistics from USA tells us that about in 2019 about 17,60,000 cases were reported and around 6 lakh deaths were also reported. Just by looking at these numbers, I'm sure that you understand why governments and pharmaceutical agencies from different parts of the world spend millions of dollars for the cancer research every year, and in the hope that they can identify better cancer drugs and better treatment for the patients. So cancer as a disease has multiple stages. As most of you might be aware of, the cancer is basically starts by the formation of a small tumor, which is an abnormal growth of cells. And as the time goes by, the tumor becomes bigger. And at this stage, actually, if the tumor is detected, it can be surgically removed and patient can recover well. However, most of the patients don't show any symptoms or very less symptoms at this stage. But once this stage passes, the next stage, the tumor gets bigger and it starts to actually invade or attack neighboring organs in this case. And that stage is basically called as metastasis. So most of the symptoms patients show at this stage, and they will be diagnosed at this stage. So that is why the cancer is deadly in many parts of the world. So now I briefly described the stages of the cancer. Let's briefly also describe about how and why these tumors form. Again, I'm trying to explain this in a very layman terms. So as most of you might be aware of, every cell in the body has information for approximately 20,000 protein coding genes. And for a normal functioning of any healthy organ, you need certain set of genes to be turned off and certain set of genes to be turned on. So when there is a change in status of some of the genes, that will lead to the formation of a small tumor. And in each stage of the cancer, you will have different set of genes changing its status, which will actually impact on the tumor growth and eventually leads to metastasis. So most of the cancer researchers basically they spend their time and they invest their research mainly in identifying actually these genes. What are the genes that are altered in case of tumor and not in case of normal? and then they identify and derive drugs which can actually target this particular genes. And those drugs are provided in the hope that they can actually cure the cancer or they treat the tumor condition in this case. So one thing that is very common is whenever they try to target the drugs, what happens is when they give it to all patients suffering from the same cancer, the same drug, it happens so often that some patients respond very well to the drug while others show no effect. So drugs will have no effect on some of the patients. And as the researchers spend more time on this concept and trying to understand why is this happening, the answer comes as this is the of tumor heterogeneity. So the concept of tumor heterogeneity and the concept of the cancer subtypes. So that is the primary reason why different people have different effect, or the same drug can have different effect on the different patients, and that tells you the cancer is complex. So if I tell the same thing in a very layman term, so the patients who have same cancer may have different tumor properties, and even in some cases, if a patient has multiple tumor, they will basically have multiple types of genes activated in different tumors. So this is the reason why the cancer is very complex, and identifying the right drug that can be used to treat all patients is very, very complex. So I hope I covered the concept here, and most of you would have now been aware of the complexity of the cancer and why it's so difficult to develop drug for it. So then let me start now with the concept of big data. So I've already, I mean, this is something very commonly used in technological terms, actually, in computational terms. But the big data has been making a lot of big noises in cancer research recently. So while explaining the biology of the cancer, I use the term very loosely that some of the genes are turned on and some of the genes are turned off. So this is not as simple as that. There are different levels at which genes are turned on and turned off. So as most of you might be aware, gene is basically a DNA sequence which can lead to different products like RNA and protein, and it can be regulated at different levels. So if a particular cancer researcher can identify whether a gene is turned on or turned off, that person has to study multiple features like RNA and DNA. And in today's age and day, now you have lots of high-end technologies to study this. So I'll probably now introduce you to the three important technologies which is very, very widely used in cancer research in last decade or so. It's microarray, sequencing, and mass spectrometry. So basically, a cancer researcher uses any one of them or all of them together to determine whether the gene is turned on or turned off in cancer condition. So microarray is basically an experimental technology which is used to detect the expression of thousands of genes at the same time. So I'll not go in depth in terms of how the technology works, but I'll tell you that the cost of experiment for one sample is currently around $400 to $700. It's about 29,000 to 50,000 rupees. And I've just also given you a brief snapshot of how the result looks like. This Excel sheet that you can see on the screen basically tells you actually how the data looks like in this case. So the next technology is actually next generation. It's sequencing, which is very, very widely used in current day research, and it's it can be a little expensive. The cost of experiment for one human sample can range from $200 to around $3,000. It's around 1,50,000 rupees. And this technology actually allows one to study entire human genome. Gene expression, gene regulation, genetic aberration. There are different aspects that one can study using this technology. And again, this is a snapshot of the result that one can see. So the next one is actually mass spectrometry, which is very widely used to detect the protein level, protein expression level. And this is relatively cheaper. It's about $33 to $200 per sample. And this is the graph that you can see is actually a snapshot of the analysis result in this case. So the objective here, why I'm showing you the results in this way, basically the snapshots of the result that you get. Now, this is actually a raw data what a computational biologist get. So most of the researchers, they spend time in procuring the samples and extracting the RNA, DNA from the sample and using these machines. So as a computational biologist, I receive these particular files. So this is called as a raw data, and it is completely responsibility of a computational biologist to analyze these results and provide to the cancer researcher the results in a human readable format, which they can make sense out of it and can take forward in their experiment. So in this case, let's see that so these are there are different steps involved in raw data processing. So once we receive the raw data, so we have to perform multiple steps that starts with data cleaning, data normalization, annotation, statistical analysis, and eventually leading to data visualization. So for those who are basically in the computational science background, so I would like to tell that most of these tasks will be done by ready-made softwares that are available. So as a computational biologist, one has to be aware of the different softwares that are available and how we can use these softwares for what type of data. So one has to be aware of it. Most of them are available with the language called as R, which is pretty commonly used. Perl, Python, and R is the most commonly used languages, the computational programming languages for the biological data analysis at this point of time. So statistical analysis generally can be done using something called as SAS or MATLAB or even R. So using those statistical softwares, one can perform the statistical analysis. And once we actually have all the data that we are looking forward to, and have the data as a visualization images, that is what we actually share with the cancer researchers. So they particularly use the information and they basically use this information and they take it forward for the next experimental analysis, or they could look for any drugs for future use. So for further purpose, they will take the results that we provide forward and use it. So this entire steps what I explained. So basically, this is basically why we call big data, because at one stretch you are looking at the status of entire 20,000 genes by using experiments. So it's a lot of data, and the file size also varies from technique to technique in this case. So I hope you are clear with all the things that I mentioned. If you have any question, we can discuss it later. So I've explained briefly about the basics of the computational biology. I've also, I've explained the basics of the cancer biology. I've also explained briefly about the different technologies that are available to study the cancer samples in this case. and what are the types of analysis that we can do it. So the next step is basically, let's go to the concept of big data. So why do we call as a big data? I mentioned earlier that with one patient or few patients, it's very difficult to find out drug or target genes to treat multiple patients. So in the year of 2005, actually 2006, National Cancer Institute in America, they actually set up a program called as the Cancer Genome Atlas program. So this is the first step towards the generation of big data from America here. So what they did was they recruited approximately 11,000 patients from different parts of Canada and US, and they collected the samples. They collected both normal and tumor samples from each of the patients, and the samples which they collected were actually analyzed using all the technologies that I described to you before. So they use those mass spectrometry, and they use the RNA sequencing, they use microarray to analyze the samples from approximately 11,000 patients related to 33 different cancer types. That's a huge amount of data that they have generated. So just before moving forward, I would like to bring to your notice that other than TCGA, there are other programs which are currently running and generating huge amount of data. So one is called International Cancer Genome Consortium. So they are actually aiming towards getting the samples from approximately 25,000 patients, and they wanted to analyze the makeup of their genomic sequence. Then there is another project called as Target, and this particular project is dedicated to understand only childhood cancers. So they recruit all the children who are suffering from a cancer, and they take a consent with them. With their consent, they actually extract the samples and they use in their analysis. So I'll now briefly explain about the TCGA, the Cancer Genome Atlas, because this is the data that we have used in our tool, You All Can, for the computational data analysis. So the Cancer Genome Atlas, as I mentioned, it's a collaborative effort. It's an effort of about 20 different laboratories in America and Canada. They actually gathered the tissues from approximately 11,000 patients corresponding to 33 different types of tumors, and used the sequencing, microarray, and mass spec technology to actually generate 7 different type of data. So in terms of the size, I'll tell you that the TCGA has produced over 2.5 petabytes of data. It's equivalent to around 500,000 DVDs. Just imagine the 5 lakh DVDs. That's the data that they have generated from the project. And in 2015, the project was complete, and they have released this entire data to public. So public here can actually access this particular data at 3 levels. They have level 1, which is actually a raw data. I showed you the snapshot of some of the raw data from each of the technologies. That's one of them. Then there's a level 2 data, which is semi-processed, which is slightly smaller in size, and one can take that and move forward. And then there is third level, which is called as segmented or interpreted data. Now this is pretty small in size, and any computational biologist can handle this particular data. So it is more handleable with your laptop or with a medium-sized server, computation server. One can handle this data. So this particular level data actually attracts most of the computational biologists for further analysis. So basically, before moving further, I would like to mention that the the project was very successful in understanding multiple cancer types, subtypes, identifying new therapeutic targets, and also the project has led to publication of about 32 different papers which reported all these findings. So with all the findings reported, should we consider that this data is completely utilized? It's not the case. So there is a huge, it's actually a gold mine of data, and anybody can actually, with a computational background, can download the data, use it further to get more insights from this data. So we, in our lab, I actually joined the University of Alabama, Birmingham in 2015. One of the first projects that I took up was the TCGA, that is the Cancer Genome Data, the Cancer Genome Atlas data analysis. So we, in our lab, when we started doing the analysis, we realized that for a computational biologist, it is easy to analyze. But what about those who have limited computational ability, like cancer researchers, doctors, who want to use this data, who want to analyze this data? There were few platforms available, but we found those are not so much user friendly. So that has prompted us to develop a new web server, which we called that time as UAllCan, University of Alabama Cancer Analysis Tool. So what we did, so now let me briefly describe about the UAllCan. This particular website is designed so that anyone can actually do the analysis. So you can Google it and you can go to the site and you can check it. It's very user friendly. So even if you don't have a lot of knowledge in terms of cancer data analysis, you can easily use this particular website to perform analysis and identify some of the important genes from different cancer and even different subtypes of cancer. So I'll briefly explain you how we developed without going into in-depth details. So what we did was, I told about level 3 data. So we downloaded the level 3 data of different types like RNA-seq, microRNA-seq, DNA methylation data for all 33 different types of cancers from TCG. And once we downloaded that, we also gathered the proteomic data, the mass spectrometry data from another consortium called as CPTAC, Clinical Proteomic Tumor Analysis Consortium. So from there, we downloaded the data, and all this data were cleaned, annotated, and processed using R and Perl scripts. So using Perl scripts and R packages, we actually processed them, performed the statistical analysis, and gathered the results for approximately 20,500 protein coding genes around in 33 different types of cancers and around 10,000 proteins in 7 types of cancer. All these results, whatever that we gathered and developed, we actually hosted in a website which was developed using Perl CGI interface on Apache web server, and the visualization features were actually developed using HighCharts JavaScript, and that's what led to the formation of UAllCan. So this is, you can see, I hope you can see, so this is actually the home page of UAllCan website. So you can use the link that is provided here, the URL, or you can also you just can type UAllCan in Google, and the first hit or second hit will be the search. It will be the website. And for those who are interested in computational stuff, currently this particular website is hosted in a Dell PowerEdge server, which is working on CentOS Linux operating system, with a hard drive space of 22 TB, RAM of 100 GB, and CPU cores of 72 CPL cores in this case. So, as to handle such a data and to handle a lot more requests for analysis, we generally need a very robust system, a server. So that's why we have this, and we also have backups of the server in cloud for the future use. So briefly, now to use ULCAN, if you go to the ULCAN homepage, and you'll be directed to something called the analysis page. It has multiple panels in the ULCAN. So each panel is dedicated to provide you with a different types of output. So if you are somebody new to the cancer analysis or study, and you just want to see what are the most important genes that are turned on or turned off in particular cancer, you can click in the links in the panel 1, and you can visualize the list of genes as a heat map. This is called as a heat map. So where you can see the level of expression of a particular gene in each of the samples. As I mentioned, there are hundreds of samples for each cancer types. So genes that are turned on here are listed in this example. And similarly, you have panel 2, where one can actually query this particular database with gene of their interest in cancer of their interest. So I'm just, for example, I'm showing you CDC25C. It's a phosphatase, so the expression of this particular gene in breast cancer. So if you want to study, so you can select in the drop down menu and type the gene name. You will be shown the expression level of the gene in different stages of the cancer, in different patient race, based on patient age, different subclasses. So there are different categories that we make from the total cancer patients, and we provide the expression information of the gene of interest in all those categories. And every information that I am showing here can be downloaded as a high quality image, and the statistics is also provided for each of the graph that we give. So in addition to this, we also provide something called a pan-cancer analysis. So what I mean by pan-cancer analysis? So you can see that a particular gene, you can excuse me. So for a particular gene, you can actually visualize expression level in different cancers. So each of them are named here by 4-letter code, but you can actually see the expression level of a gene in different cancers in one shot. Pardon. So we actually published this particular database in 2017, and recently when we actually included the proteomic data, it was published in Nature Communication Journal. And as it was mentioned in introduction, I'm very glad to tell you that this particular tool is used worldwide. So we have nearly 4 lakh accession. So people from America, China, Europe, India, so all of them actually have used this particular tool for their cancer research. And we have over 500 citations also for this particular database. So basically, citations here basically means that they use the tool for their cancer research and they cite us. So we are coming to the end of the talk. So basically, this is a snapshot of the city where I live, Birmingham. It's the southernmost part of the America. So I actually work in this place called UAB Comprehensive Cancer Center in Birmingham. It is located exactly adjacent to University Hospital, and we have more than 50 different cancer researchers working here on different types of cancers. Like some of them are working in breast cancer, some of them working in ovarian cancer, and some of them working in other type of cancers in this case. And finally, I would like to thank my fellow mates. This is the photo of my lab mates here. So for further help in developing this particular tool, and also thank my guide, Dr. Suri Varamballi, for his constant support. Thank you. I would like to take any questions that you have with regards to this.
If any participants have your queries, please drop a text in the chat box so that Sir can answer a few questions. Dr. Darshan sir, it was really an awesome and informative session. The union of computers and biology has really made data analysis very easier and very effective one. Thank you so much for joining us in this.
Thank you very much. Again, I would like to thank the organizers for providing this wonderful opportunity. And it's absolute delight for me to speak to the students and the faculty of my own college. Thank you.
You're indeed our proud alumni, sir. Okay, now let's move on to the next session. Now I welcome Netawati ma'am, Head of the Department Computer Science, to introduce the speaker for session 2, Dr. Nagesh K.A.
Hello everyone. I would like to give a brief introduction about Speaker 2, Dr. Nagesh K.A., Senior Research Investigator at Syngene International Limited, Bangalore. He completed his BSc degree with ZBTCS combination at the DVS Degree College of Arts and Science, Shivamogga. He pursued his MSc in the field of biotechnology from Mangalore University, and he awarded PhD from Indian Institute of Technology, Roorkee in 2012. He had done his postdoctoral fellowship at C Camp, Bengaluru from 2012 to 2014, completed genome and transcription sequence analysis of leaves, worked on raw data, filtration and assembly of Tulsi genome. He worked as a scientist at Genomic from 2014 to 2017, and presently he is working in Syngene International Limited, Bangalore as Senior Research Investigator. I am very happy that he is an alumni of our college and guest speaker for today's webinar. I welcome Dr. Nagesh K.A., Senior Research Investigator, to handle the session. Thank you.
Thank you for introducing the speaker, ma'am. Over to you, sir.
Hello, everyone. It's a wonderful opportunity to come back to the college and speak to the students there as well as my teachers for a long time. And it's been a nice opportunity for us to interact and as well as understand how the field of computational genomics is being taken forward. And I would like to take this to thank the organizers of this webinar for providing such a good opportunity to share and discuss the scientific methodologies that are being used in computational genomics, both in academics as well as in industry. So the talk will be basically split into basically 3 to 4 sections, where I would like to start by giving a background of what genomics and next generation sequencing about. I saw Dr. Darshan mentioned next generation sequencing in his talk, and I would like to slightly go in brief detail about the technology of it, and then a general method of analysis, that how a scientist would plan a data analysis and how would you approach a specific set of data from raw data to, let's say, the kind of results that we are trying to look at. And I would like to go with a few examples of data analysis that I would do. I could take up in different areas over the few years of my work in this area. So to start with computational genomics, as you know, it is an interdisciplinary science, and it basically involves the data analysis from DNA and RNA sequence. When I say genomic data analysis, it's mostly derived from DNA, RNA, and the metadata are the allied data for that source of the sample. And this field basically got accelerated with the developments in both computation as well as in biological research actually. So as Geeta ma'am mentioned in her introduction, so this whole field of computational genomics started with the completion of, or with the efforts to sequence the complete human genome in the last century, and it was completed in 2003. And later on we have seen many developments since then, because we realized that we need to sequence more and more human samples, as well as we wanted to sequence all the other species like plants, animals, insects, bacteria, everything we wanted to sequence to understand how we relate to them, and also to derive practical applications from the data set and gene diversity that is available in different organisms. So this is how you can say the field has evolved. So this is just basically a cartoon explaining how difficult it was to sequence a small fragment of gene maybe like 20 years back, where you may have to take one sample piece of DNA and you will be able to sequence it in maybe a half a day's work, and then that would basically give you something like 500 to 1000 base pairs of DNA sequence. From that, scientists they evolved into the next generation sequencing, where you can take up sequencing of entire genome or entire transcriptome of a sample in a single shot. That basically means, an example like, instead of fishing for a single fish, you basically go and take a motorized approach and fish a huge amount of fish that is available in the water. But this approach also enhanced the availability of sequencing, and more development started happening in terms of improving the efficiency of sequencing. And that's where you can see since 2006, the start of next generation sequencing era, the cost of sequencing a genome has started falling drastically. It has overtaken the Moore's law, which basically applies to computation, where you can say that every 1.5 to 2 years, the cost of the computation capacity almost halves every 1.5 to 2 years for the same architecture. But whereas you can see that the chemistry of sequencing has gone much faster than the speed of computing, and that's where we have to understand the data better, and you have to use the computational resources much better. So in terms of sequencing, we can divide this into two major categories, like first generation sequencing, which is basically predominantly Sanger sequencing, which most of you might have studied in your syllabus, and then next generation sequencing, which you can say there are sub sections of it, like second generation or third generation sequencing, where second generation sequencing mainly covers data sets that provides short fragments of DNA. That means it can sequence somewhere between 25 bases stretch of DNA up to like 500 to 600 bases long reads of DNA, but it cannot go beyond that in a single read format. Whereas the development in the third generation sequencing that happened in the last few years actually developed into what we call it as third generation, where we can see a lot of long read technologies coming up, where you can sequence few kilobases or even megabases length of DNA in a single stretch of sequencing. And these are the sequencing instruments that are available in the market. You can see that on the top left hand side, where you can see MiniSeq, MySeq, NextSeq. These are all sequencing instruments from Illumina, which is one of the most prominently used sequencing system. And on the right side you can see Ion Proton. On the below you can see like PacBio and MinION. These are all the sequencing instruments. The below ones are third generation and the above ones are second generation sequencing instruments. And the main role of these instruments is to do DNA sequencing in parallel. That means instead of sequencing one fragment at a time, you sequence millions of fragments at a time in parallel. So that is the power of next generation sequencing machines. And to come up with a basic understanding of Illumina technology, which is the most widely used now, it basically uses a flow cell based DNA sequencing system, which also has a unique sequencing by synthesis method, where you prepare your samples by cutting this DNA into small fragments, which generally ranges between 150 bases to around 600 to 700 bases. Then a molecular biological method is used to attach a known sequence of adapters on both the ends, and then you can basically allow it to run it on the flow cell, where a sequencing chemistry happens, and where a small DNA fragment is bound to the flow cell based on the complementary DNA sequence that is available there of the fragment DNA, and that will actually help us in making a isothermal amplification on the glass slide, leading to a cluster of DNA which will be sequenced base by base for us to give the sequence information. So if you see this, when you see the Illumina sequencing data in the crude format, you basically see a glass slide with some bright spots, which is the fluorescence from each of the clusters that is coming up, and an automated base calling algorithm looks at each fluorescence at each base that was pumped into the instrument, and it will basically give this output in the form of a FASTQ file, like Dr. Darshan gave an example before. So this whole sequencing chemistry is very much modifiable in a way that you can basically select any of the targeted region that you want to sequence, and also you can also choose how you want to sequence a specific target region or an entire genome or an entire RNA of the cell. And that basically becomes a question for many of the scientists who is working. It's like a menu of the restaurant, where you have to go and select what is your sample and what is the kind of questions we were trying to address, and what kind of methodologies we want to use for it. Let's say if you want to understand the whole gene expression of a specific tissue, let's say cancer, you basically want to get a transcriptome sequencing done. Whereas if you want to understand the microbiome of the gut, that is what and all the microbes that is available inside the human gut and that is affecting our biology, then you may want to go and do a metagenomic sequencing. And if there are certain areas, like if you want to look at if there is any epigenetic changes in a certain system, you can look at methylation sequencing. So like that, you have different sequencing methods to interrogate different questions of biology. So what do you get after the sequencing? At the end of sequencing, you will be ended up with a huge amount of data. And that data basically will come in the format of a FASTQ file majority of the times. And when we say this is a huge amount of data, it means that a single sequencing run can a range between a small run of 5 GB data to a terabyte of data in a single sequencing run, which can take just few days to sequence. So that's where the role of computational biologists come in, to see and understand this huge pile of data which comes in the raw format. So what are the basic skills that a computational genomics scientist would like to learn or would have to do these analysis? So the basic understanding of bioinformatic applications, that means we need to know few of the major applications or most commonly used bioinformatic applications that are used in the field of genomic data analysis. We should be able to understand the algorithm or the methodology by which this application works, because you cannot just run the program without the understanding, as it leads to false interpretation. We should be able to compare different applications and pick and choose the right application for the right analysis. And there are sometimes where you end up with certain situation where one single tool cannot give you the result that you are expecting or the interpretation that you are looking for. So you have to figure out and organize different applications in an orderly manner to develop an analysis pipeline. That is, I'll do the analysis with application 1, the output of that application I'll be giving it to application 2, and so on. If there are certain parameters where you want certain things to be analyzed but you don't have any known application or validated application, at that time you can also program your application for the new methodologies you are trying to derive it. And you can choose any of the programming languages for this application. Most of the people nowadays are using somewhere from Perl, Python, or even basic shell script is also working well for many of the applications. And if there is no limitation, you can even use C or Java wherever you are comfortable with. And at last, you also need to have some biological knowledge to understand and interpret the data, or else you need to collaborate with a biologist to get that biological context of the data to interpret this data better. So this is how a data looks in a FASTQ format. You basically have a header information where the sequence header is mentioned, then you have the sequence, and then you have a plus which basically separates two lines, and a quality score line which says what is the quality of the data. And all these information is useful when you go with any analysis. So when you start with a basic analysis of next generation sequencing data, we want to see what is the quality of my sequencing before doing any interpretation, because if I have a bad quality of sequencing run, we should not be using it for any of the interpretation, and that is one of the key parameters before we go ahead. And we can also see that whether the data is single end or paired end, where each kind of data has to be processed in a different manner, because if a single end data is there, it basically has a small fragment of DNA being sequenced in 5' to 3' end, and that data is much simpler to handle. Whereas there are most of the Illumina runs are paired end, where you will have both forward and reverse read are being sequenced from a fragment of DNA. That means to say that if you have a sequence fragment from 200 bases to 1000 bases, and you are sequencing, let's say, 75 bases on each end, that means you know that one side of 75 bases and another side you know 75 bases, there is also an unknown sequence in the middle. So these are the kind of data that generally a next generation sequencing machine gives out. And when we want to understand how the quality of this data looks, what we do is we take each base position for all the reads in that file and try to compute what is the base quality at that base position for the entire file. Because if you say in this example, you can say that we have around 520 million reads, and you cannot really quality check and understand the quality as a whole unless we format it in a reasonable manner. And here you can see that on the x-axis is basically base position, base number 1, base number 2, base number 3. On the y-axis we see the quality score. It is kind of a log data where whatever the value we see is basically the interpretation of the quality is what is the percentage, what is the kind of error we can see in the data. So if I say that my quality score is 20, that is translated as 10 to the power of 2, which also means that one error in every 100 bases sequenced. And if you say if my quality score is 30, which translates to one error probability in every 1000 bases sequenced. So when we see data like this, we basically try to minimize the errors in the data. On the left side, the file looks quite reasonable, and you can basically keep most of the data. Maybe few reads can be eliminated based on the quality. Whereas on the right side, you can see that the data actually falls off at the end sometime for certain reasons, and you may have to do what we call it as trimming. So you will take this sequence and say that I don't want to keep the sequence beyond 100 bases or 105 bases because I see a drop in quality. So this is the kind of quality filtration we can see. So now when we have a quality filtered data, then how would we go about approaching this data analysis? So your data can be analyzed both de novo as well as reference based analysis based on the kind of data or the background of the experiment. In an example where you want to go with the de novo analysis route is where you don't have the reference genome of that sample. That means we don't have any background information. Let's say we are sequencing a new bacteria or a new plant where we don't have any database for which we have any genome data. You have to go with a de novo assembly approach. And then with the de novo assembly, we can go with gene prediction, gene annotation, and also if we have sequenced the RNA, we can also look at transcript data and so on. Whereas if you have the reference database for a given sample, you can basically use this reference to align our data to that reference genome or a transcriptome, and thereby start to interpret in reference to that genome how our data looks, where our data varies in comparison to reference database. Let's say if there is a single nucleotide polymorphism or if there is any small insertions and deletions, we can look in reference to that data. We can also see the transcript expression in reference to the reference database by saying that my gene of this annotation is getting overexpressed or this gene is underexpressed. Whereas in de novo, you will basically identify each gene and try to annotate yourself. So let's go with examples for in certain de novo approaches as well as reference based approaches and try to see how these data analysis is done and try to interpret it in a way. So this is an overview of a genome sequencing project of NEEM, where we had generated different data sets, and you can see in the orange boxes what are the outputs of the analysis, and you can see in the blue colored boxes the programs used for such processes, and also you can see on the lines what did that program do for us in that whole process. So we start with the sample here, where you can say that you have a tree of neem where you collected leaf and extracted genomic DNA. You also took few other tissues from the plant, you did RNA isolation and you took it for RNA sequencing, and you also took this data for metabolite analysis to know what kind of metabolites this plant is synthesizing. And when it comes to the DNA sequence, it goes through an approach of de novo assembly, because when we were working on it, there were no reference genome available in public database for this tree species, and hence we started by assembling the genome. The assembly of the genome is initially fragmented, and more and more sequencing will be added on to get a better scaffolding of the data. And where we can see that the genome assembly was done using a program called Velvet, which is basically a short read assembly program which uses a K-mer based alignment strategy to assemble it. So the way it works is you take a small read, you cut it up into small pieces. Let's say if you have 100 base per read, you take the first 25 bases and try to search in the entire data set if that 25 bases is found anywhere else. And if that 25 base is found anywhere else, you take those reads also together with it and see whether it fits in order one next to another. That's how these assembly programs work, and you can get a genome assembly out of it. But once you get the genome data, it doesn't really help us in any format with just the sequencing. So you need to do multiple analysis of it to find out what are the genes in it. So what genes in it is difficult to find without any known information. So what is the known information that we are going to feed in is, we will feed in a model of Arabidopsis or tomato or other plants, where a model is made to basically train the system to figure out what a gene looks like, and that program basically helps to predict what is the genes of that sequence. And the way we validated these genes are true is using the data from RNA sequencing, and when the RNA sequencing data is aligned against the set of predicted genes, so you take the RNA sequencing reads and you compare with the genes that you predict, and whatever actually matches with it, you are sure that those genes are real and the plant is using those sequences to make proteins as well as other RNAs. We also use different programs to identify what is the repeat content polymorphisms in that data actually. So you can say that we use different tools for each of the purpose, like to identify repeat, you use a repeat modeler, you identify SNPs and indels, you use VCF tools, and to identify simple sequence repeats, you use a program called MESA. And the genes that you have will also be useful if you can give much more information to the public with this. So how would you enhance the information for the gene? What we basically can do is take the gene sequences that we know and try to align it with the known genes from other species, let's say Arabidopsis or rice, wheat, tomato, whatever plant species or other species we have, you can align these sequences using the databases of those. So in this example, you can see that we use different programs like BLAST, Go Enrichment, KEGG pathway, and Interproteuscan to annotation of each gene. So to identify what is this gene and what are the similar sequences in other species doing in them. That gives a better understanding of what those genes are doing, and we get a set of data annotations. While doing all these things, there are certain areas where you can see we can clearly fit in our tools that is publicly available, which is developed by scientists already. But there are certain areas where you may not find a tool that you are looking for. And at that time, you can start developing your own tool as well, and that can become a resource for other people as well. So this is one of the pipeline that we worked on that time, where we wanted to identify simple sequence repeats that are different between two plant genomes that we already sequenced, but the genome was in a draft format. So this pipeline was developed using a combination of Perl and R scripts, where the initial process of raw data identification and filtration happened using Perl, and the data was aligned between them using bowtie, and the data output that came out was again filtered and identified to find out what is the polymorphism using R script. So this is how you can mix and match available tools and coming up with your own tool of your requirement. And when you go for validation, this is what you would like to see a good tool performing. So the idea was, you can see that in the first sample you can see there is a 4 base deletion, whereas other 3 samples those sequences are intact. And these kind of genetic variations are useful for us to mark certain genomic regions and known as markers for many of the plant-based studies actually. So for that application, this whole pipeline could identify many of those targeted simple sequence repeats which were separately annotated and made as a database for the neem genome researchers who want to study further on the neem tree. So this is one of the examples where you know how you take the data and interpret with different kind of softwares and applications, and there are cases where you don't have, you go and build your own. So if you have any doubt or a question in this section, you can ask me, or else I would like to go with another example and try to explain you how another data set looks. So there was also another project where we wanted to study a breeding population of melons, where there was a need to transfer one of the traits in one plant to another, and this was basically a seed industry requirement, where you have an high yielding plant and you want to transfer a disease resistance from another low yielding variety, but want all the agronomic traits of a high yielding variety. So for that you need to identify what are the regions of the genome that are differing between these two samples and find out what are the regions that are responsible for this disease resistance. So here you can see an example of that kind of an analysis, where we took a breeding population where 2 parents were sequenced and also the progeny F2 population was sequenced, and a phenotyping data was done to figure out what samples showed the resistance for the disease in the field area. And when you sequence those samples which had resistance versus other data, you can see clearly that there are specific genomic regions where you can see either it is heterozygous or it is derived from the donor plant which basically had the resistance for it. And these regions that we mark in these experiments becomes a plant breeding markers that can basically trace the trait of each individual plant with that trait. So for example, if I know that this is the region involved in one of the disease resistance, and you can basically develop a PCR based method to detect whether this region is there in that plant, to call it a disease resistance plant. Now if you want to go ahead and want to merge two different disease resistance or three different disease resistance in the same plant, then these kind of markers will be the detectors of those plants which carry those disease resistance genes in that plant, and that helps in developing new plant varieties that are much more disease resistant, more and more yielding, and also benefit for all the farmers that actually grow that variety. So similarly, you can see this application in human as well as animal health, where you can see this is another example where we looked at data for a toxicity of an experiment. That is, we know there is a new molecule as a food additive or a food ingredient, and we may want to know whether that molecule is toxic to humans or animals, and if it is toxic, at what concentrations, because any molecule given at a very high dose is obviously toxic, but we need to know whether it is in the safe consumption limit for humans or else animals. So this is where this experiment you can see, where a scientist actually fed these compounds to rats for different periods like 7 days and 28 days, and the rat liver was taken to do a transcriptome data sequencing. So you sequence all the RNAs that are being expressed in that liver tissue at that specific given point in time, and you also have healthy control animals where you can say that you have all the body parameters are healthy, and you see how different genes are being overexpressed or underexpressed at different treatment conditions, and you also try to see whether a 7-day treatment versus a 28-day treatment, if there is any difference. You want to see whether there is a difference in males versus females, and all this data are put together as a format, as a heat map, to see that what genes are overexpressed and underexpressed. So what this means is that with this data, you can see that there are certain genes that are getting overexpressed, certain genes that are getting underexpressed, and you can basically use this overexpressed and underexpressed genes to model the data to see what pathways this compound is going to affect and what could be the potential limit of toxicity for this compound. So let's say you have this data available. If you can model it, what it would basically say is that certain metabolic pathways and homeostasis is affected by this compound, and if we look at different dosage conditions and different treatments, you can see that the change in genes can be really significant after a dose range of around 750 mg per kg per day, whereas the dosage the gene expression changes starts with around 500 mg per kg per day. And this kind of interpretation really helps industries to evaluate their compounds even before they can actually invest in developing this molecule and do a lot of human trials or animal studies, and try to reduce the risk for the population as well as increase the speed of development of these molecules. Similarly, these kind of applications can be used for different questions, and each one of them may have to have a different thinking process. Even though we may have the same tools and computational methods, the scientist or the computational genomic expert basically looks into all these parameters and chooses that kind of tools these systems provide us to understand these biological data in a much better format. Now we know that we can use this in various kinds of scenarios. Let's say what is the future, how we can even improve this further. Let's say you are in college now, maybe in a period of few years you will come out and you want to do some kind of research, and where the field is headed. To say that, you can say that the sequencing cost is obviously reducing, and also there is a drastic reduction in the cost of gene synthesis, and also development of new methodologies like sequencing of single cells. Like we can see lot of heterogeneity in terms of immune cell population or cancer cells. So if you are able to separate a single cell separately and you can sequence them, then you can get a better understanding of the heterogeneity of the population, and that kind of data will also have a multidimensional analysis requirement, and that is where many of those new applications that are needed to be developed. There is also a lot of automation and robotic systems that are being used for biological experiment. So there is a lot of requirement of computational knowledge as well as a biological experimental design to develop these automation approaches for doing these analysis. And also there are systems biology approaches with machine learning and artificial intelligence, where people are trying to put not only genomic data but all the available data for a specific sample to see whether we can develop a model in the in silico system which almost simulates the biological component of it. So this is how the field is being explored further, and I hope many of you get interested in this kind of research and would help them basically improve your knowledge in this area. So this is what I was trying to cover in this session. I would also thank the organizers once more for giving me this opportunity, and I can take any of the questions that you may have.
Well, that was an amazing session. How amazing it is that using computation and statistical analysis, computational genomics deciphers the genomic sequences, including RNA and DNA. That's really amazing. Thank you so much, sir, for being a part of this webinar. And indeed, your college is really very proud of having you as the alumni.
Thank you all.
And now it's time for a thank you note. Feeling thankfulness and not expressing it is like wrapping a present and not giving it. Now I request the member of College Alumni Association and also HOD of Botany, Professor Sudhakar sir, to propose the vote of thanks.
Good morning everyone. As the part of Alumni Association and faculty of this institution, I feel honored and lucky to have this opportunity to propose vote of thanks for one-day international webinar on prospects in computational biology. It was a great pleasure to have the alumni of our college, Dr. Darshan S.C. and Dr. Nagesh as the distinguished speakers of today's webinar. On the behalf of DVS management, principal, staff and students and organizers of this webinar, I extended my heartfelt thanks to these eminent speakers. I would like to thank our DVS management for their constant support for all the activities of the college, and special thanks for Secretary Rajashekar S. and Director Dr. Satish Kumar Chetty for addressing today's webinar. I thank for respected Principal Dr. H.P. Krishnamurthy sir for his presence and guidance regarding this webinar. I also thank Dr. H.V. Anil Kumar sir, IQAC coordinator for this college, for his constant support in organizing this webinar. I extend my thanks for Dr. Geeta Shamat, Head of the Department of Zoology, and Ms. Netravati H, Head of the Department of Computer Science, and members of Alumni Association for organizing this wonderful one-day international webinar. I wholeheartedly thank ICT Coordinator Kumar Swami and IT Cell Technical Team Megharaj, Jagdish A.S., Akshay Prabhu, Sangeeta, and faculties of Computer Science Department of our college. I thank. I also thank to Mr. Jagdish and special thanks to Mr. Jagdish, Akshay Prabhu and Sangeeta for hosting and moderating this webinar. I thank the non-teaching faculties of the college who helped us in various ways in arranging this webinar. Last but not least, I thank all the participants of the webinar who actively made this webinar successful one. Thank you.